I'm actually standing in for Dr. Jo Ireland, who has led this initiative when she was based at the Animal Health Trust. She's since moved, fortunately, to the University of Liverpool. So much of the work that's gone on here is directly attributable to Jo. So as James said, we're talking about equine grass sickness. Maybe not everybody in the audience understands what this is, but this is a severe neurodegenerative disease of horses. So it affects the nervous system, particularly what we call the autonomic nervous system that controls things involuntarily, and particularly affecting, as it happens, the gastrointestinal tract. Carries a high mortality, probably in excess of 85% of these cases will unfortunately be fatal. And that's especially where we've got acute and subacute, and that simply describes the progression of this disease, and usually that's within a week they will be dead from this condition. This was first recognised in Scotland, actually, the northeast of Scotland in the early 1900s, and then over the next 20 or so years it spread down into England and into Wales. And Great Britain is still recognised as having the highest frequency of this disease. It is seen in South America, a disease called mal seco, and it is seen in other parts of northern Europe. But interestingly, we don't hear of many cases reported from Ireland or indeed North America. The national average here is about 2 cases per 100 horse years at risk on affected premises. So this will be a concept that you hear a little bit more about. So essentially what this means is if I've got 100 horses and I keep them for a year on one or more premises, I would expect on average to see about 2 cases of this disease. So that doesn't sound very high frequency, but obviously if you're on the premises and that's recurring, then you are going to be losing stock because of that. So back in 2008, we established, through the funding from the Horse Trust and with the help of the Equine Grass Sickness Fund, which is a charity dedicated to this disease based up in Edinburgh, we developed a national surveillance scheme, and then we both looked back and forwards, and we gathered 10 years worth of information about this disease and what was going on. So in that data set, we had about 1,400 cases of this disease. We knew their location by country in just short of 1,000. And what you'll see here is that we actually numerically see more cases in England than we do in Scotland now, but the density of horses will be different such that it's still, seen in Scotland amongst its population. Very few cases in Wales. So these graphs are actually taken from that paper. This describes the seasonal distribution by month, and this is pretty repeatable most years and in most countries where it's seen, in the United Kingdom. So we often see this peak in May, obviously starting to go up in April, starting to come down in June, but every month of the year we will have this disease diagnosed. The data that we had from this paper showed this very intriguing sort of wave pattern of disease. Now what we're not sure is this representing some true pattern within disease, which it could be, or is it just that we've got a bias because we started collecting data and we looked back there? So that's something that we're keeping an eye on. This is the age distribution that we see, and here we see what we call the median. So if you line all these ages up, the one right in the middle is about 6 years. The mode is the most frequent one, that's about 5 years. So this is predominantly a sort of young adult horse disease, but we do see it starting to tail off. And we do see cases being reported in older animals. So it's not just a young horse disease, but it is predominantly a young adult horse disease. So that was for period 2000-2009. We've recently looked again at the data that we've accrued since then, up to the end of 2017. Fewer cases actually reported into the surveillance scheme for that period. And I'll give you the figures in red, how they contrast with the period 2000-2009. So this again is the geographical distribution. You can see that actually the number of cases being reported from England has gone up proportionally, slightly down in Scotland, about the same in Wales. So whether that's a true change in geographical distribution or it's just a reporting bias, again, we don't know. The gender split is absolutely the same as it was for that previous period. About a little bit short of a half in mares, a little bit short of a half in geldings, and then stallions about 6%. These are the different forms of the disease. The acute, so these animals will, between 2 and 5 days, they'll be sick and being put down very often. Subacute, a little bit longer, and chronics, they will last and a proportion of them will survive. No change in that distribution. The age, again, median age, absolutely the same. We've got a slight change in the proportion of animals surviving, so that's encouraging. There's some really good nursing developed at the University of Edinburgh, and where that's applied, it's showing good results. So let's bring this back to the vaccine trial. So obviously the majority of cases, the clue's in the name, they're at grass, hence the name grass sickness. So it does suggest that the potential cause is either present in soil or at least at pasture. The early post-mortem studies that were done on this condition did seem to isolate a bacteria, and here we are again, Clostridium. And it was similar in appearance and toxigenic properties to what was then known as Bacillus botulinus, but now we know as Clostridium botulinum. So the hypothesis was hatched many years ago, many decades ago now, that this was what we call a toxico-infectious form of this infection, and that was the cause of equine grass sickness. As we've heard from Laura, C. botulinum is present in the gastrointestinal tract, and the hypothesis is that a combination of the different risk factors that come together on affected premises at high-risk times of the year in potentially high-risk animals is that leads to toxin production locally, and that's what causes the neurological damage. So, the botulinum theory, we can go back to 1922-23. There was actually a vaccine trial, so we are reinventing the wheel. We're not inventing it, we're just reinventing it. And that was conducted in Scotland and involved over 2,000 horses. And that's a good-sized trial. On every premises where it was conducted, half of the horses received what they called an antitoxin neutralized botulinum toxin vaccine. So they had the toxin and then they neutralized it with blood that had been raised against that. So that was their vaccine, and the other half of the animals were actually left unvaccinated as controls. And we now realize this is a really good design in that you're controlling for the effects on each individual farm by splitting up the vaccinates like that. They actually did see very significant reduction in mortality due to grass sickness, and this was published. But the veterinary profession being what it is, not everybody accepted this. It was actually rubbished, and the vaccination against grass sickness was dropped, and it's kind of dropped out of history. Well, that is until 1990 when the theory was revisited, and these are some data that were created by Ian Poxton and his PhD student Leonie Hunter in the late 1990s. And what they demonstrated was that where you compare controls that have a low proportion of these non-affected animals have the presence of this toxin in their guts. And you compare that to all three different forms of this disease, and these cases all had, or not all, but a large proportion, they were able to demonstrate the presence of this toxin. And also, where this toxin was found, it was found in higher levels in the diseased animals than in any of the controls. So this obviously stimulated some research interest, and the same group were able to demonstrate that these cases also had low levels of antibody, therefore immune protection against these toxins at the time. This was then followed by a nice piece of work that was conducted at the University of Liverpool, and Helen McCarthy's PhD, and what they were able to show was that the risk of grass sickness decreases according to how much antibody you have against these toxins. And this followed what we call a biological gradient, which is really important when you're considering causation for disease. So what do we mean by this? Well, these figures here, which are expressed as percentage, you'll see that they go up in value, and that means that in each category there, you have an increasing amount of antibody against these toxins. And compare that against what we call the odds ratio, and this is just simply comparing everything back to this baseline group here. So you divide the odds of disease by that, and you'll see that these values are less than 1, and they go down, decreasing less than 1 as we go. So as the antibody increases, the risk of grass sickness decreases. So what this tends to suggest is that if we can artificially stimulate that antibody, perhaps we have an opportunity to protect against this disease, grass sickness, which we believe is caused by presence of this toxin in the gut. And there are actually obviously precedents for this, linked to botulinum, or linked to Clostridium at least. Obviously we have Tocker's work from the 1920s. You'll all be familiar with the use of tetanus toxoid vaccines. So again, a different Clostridia causing a different clinical problem, but a highly effective vaccine that we give routinely to horses, or at least we should be doing, in order to protect them. Highly effective vaccination. There's a condition in America called Shaker-Foal syndrome, and that's caused by a toxico-infection, again within the gut, caused by a different type of botulinum, this time type B, and this is available as Botvax-B and is used successfully over there. And also looking at other species, but the same type of toxin, type C. They are mink are susceptible, cattle are susceptible as well, and both those species benefit from use of a type C vaccine. And in fact, it is the mink vaccine that we've adapted for use in our trial. So coming back to the trial, the randomized control trial is considered really the best tool to go out into the field and demonstrate effectiveness from vaccination. Ideally, we might have an experimental challenge model that we can reproduce in experimental animals, and that's what we actually do to demonstrate the efficacy of things like flu vaccines. Limited number of animals, we can expose them to flu, we can test the vaccine that way, and we can demonstrate that it's highly effective, and we can have the products licensed. But people have tried, and unfortunately there is no experimental animal model for this disease. It's complex, and we know that from the number of different risk factors that there are for it. So I'm afraid the field trial, taking this out into the field, demonstrating effectiveness out in the field, is the only way that we could really make progress here. So, we think it's a great idea. Tucker did it in the 1920s, but there's quite a bit of work you have to do before you can take this out into the field. So, the first bit of work was a preliminary safety study performed in horses. This was done in the United States under commission from the manufacturer, taking the mink vaccine, putting it into horses. This confirmed the need for 3 doses in the primary course, the same as is seen with Botvax-B. It demonstrated good safety under experimental conditions, and it did demonstrate, actually, I think here you can see these blocks gradually rising. This is evidence that immunity, antibody is being stimulated in the vaccine group, but is not being stimulated in the placebo group. So all these data were put together, and we got an animal test certificate in order to take this vaccine and use it in the next stage, which was going to be a pilot field trial. Joe also conducted what they call a feasibility study. These are recommended certainly in the human field, and they allow you to refine and get early feedback on the benefits and how you might be able to conduct these trials. And Joe did that. This work was published in the Veterinary Journal, and these feasibility studies, which were entirely questionnaire-based, helped inform the design of the final study. So I mentioned the pilot field trial. Again, really important to understand how this might work when you scale it up. And this work was conducted in 2012, 2013. We had 5 participating veterinary practices in East Scotland, 10 participating equine premises. Recruitment for this was completed in 5 weeks. That looks encouraging. And the feedback from all of this did mean that we could improve the design of the final study. This confirmed that this vaccine was safe under field conditions. We had no reports of adverse reactions. And the majority of the vaccine group did show significant immune response. So this graph here, you'll see that we've got a small box here and a larger box here. I've drawn a line there. That's the ratio that is set at the value 1. We take 2 blood samples, one before vaccination and one following vaccination. We measure the antibody. If we divide one by the other, where we get the ratio of 1, that demonstrates we've got no difference between those 2 levels. The vaccine hasn't had an effect. But where we have an increase over 1, in other words, the amount of antibody after vaccination is greater than when we started at baseline, then that's strong evidence that we've got a vaccine effect. And that's what we see here. This is the vaccine group. Most of that data is above this line here, and you'll see that the placebo group didn't show anything there. So something is going on with this product. So that brings us on to the trial. So we decided on a randomized, placebo-controlled, triple-blinded, so the owners, the vets, and actually the participants in the facilitators of the trial itself didn't know. Only a limited number of people knew what the coding for the vaccines were. We set out to try and find 1,000 horses or ponies that we could recruit, and we targeted premises on which we knew there was a high frequency of disease. It was important for this that these horses remained on the premises where they were and under the veterinary care of their own veterinary surgeons, and that's how we conducted it. So this is the trial design, very simple. We have our eligible horses, and then we randomize them just using random numbers to receive either vaccine or the placebo. And that's done on each premises, and we also did what we call stratifying by age. You see that we have more cases amongst younger horses. So animals that were 8 or younger, we randomized in one set. Animals 8 and older on each premises, we randomized using a different set. So random allocation was a 1-to-1 ratio. So for every placebo, we had a vaccinate. And as I've said, we stratified by age as well. So what did the trial look like by numbers? Well, we had 558 horse owners that were assessed for eligibility. We had just over 1,100 animals that completed the initial enrollment forms, and of those, just over 1,000, 1,029, were actually randomized finally to treatment groups. The first horses were formally enrolled in the week, within 1 week of launching the trial in March 2014. But as became evident, recruitment was slower than we had anticipated, and we only recruited our last horses onto this trial in May 2016, or rather our new horses onto it in May 2016. So in the end, we ended up with 88 participating veterinary practices, 224 participating horse owners, 120 premises that met the criteria. And we closed recruitment of new sites, although not obviously to new animals, in September 2015. Just over half of these were in England, just under half were actually in Scotland, and these data here are represented across 44 different counties in Great Britain. So I mentioned about the frequency of disease that we had measured from the surveillance scheme. Well, on these premises, we had just over 2 cases per 100 horse years at risk. So we knew that we were targeting a representative sample of sites. So this is really a summary of the treatment phase. So we had our primary treatment course and then follow-up boosters after that. We'd initially planned that we would only need 1 booster, but as became evident, we needed to extend this trial longer and we had to add in another booster. So essentially, these are the injections of vaccine or placebo. We took blood around the time of the first sample and at the end of each course. That was for health monitoring purposes and also to be able to measure the responses to antibody. And after each injection, we required that the owners carefully check their animals, keep an eye on their animals, and report back any suspect adverse reactions. And if the vet needed to be called, then that obviously would be covered by the project. So the first trial injections were made in April. The last booster was completed in halfway through the year in 2017, and then we followed those animals up to the end of 2017. So that's when the trial finished. Detail in this slide is not important, but as you might understand, when you're doing these studies to meet requirements for licensing and VMD, there are a lot of paperwork, and so there were a lot of questionnaires, a lot of forms to be filled in and got back and kept track of by the team. So what was our trial population in the end? Well, they did in the end contribute more than 2,000 horse years at risk. So doing the simple math, feeding back from the frequency that we thought, well, that should have given us around 20 grass sickness cases. And that's our original sample size told us that that's what we needed. The median horse age at enrollment was about 8 years. The distribution of genders, sexes, was about what we saw in our surveillance scheme. These ranged in size from miniature Shetlands all the way through to large Irish draft crosses. We had everything from companion pets to national competition horses, so a broad range of animals were represented. When we've looked at this population, we found no difference between the 2 treatment groups with respect to age, sex, or breed, and that thankfully confirmed that our randomization approach had been successful. As with any trial, you're always fighting against loss to follow-up or loss of animals out of your population, and that indeed was the case. So even after we'd recruited what we thought were animals, to getting them to the treatment being started, we were even losing animals then. Horses have a habit of moving premises, and indeed we did unfortunately suffer from that. And even amongst 974 horses that received an initial vet visit, only 97% of them, not all of them, completed that 3-dose primary course. 76% of horses went all the way through to receive an annual booster, and then about half of that group went on to receive another booster, which initially we hadn't planned for, but which we needed to put in place in order to complete this trial. So, what are the results so far? Well, thankfully we had a very low incidence of adverse reactions. So this slide here, you may not be able to see, but that's a big swelling on a neck where a horse has been injected. I would urge, this is a Google image, it's not actually from our trial, but it's just to illustrate the point. And where we did have those few veterinary attended adverse events, they were not all in one group or the other. So injecting horses with anything into their necks does run a risk of adverse reactions. So what about the cases? Well, we had 9 grass sickness cases diagnosed during the trial. One of these actually occurred after a horse was technically excluded as she became pregnant, and as James said, we were excluded from conducting this trial in pregnant mares. So our overall incidence that we resulted in was 0.4 cases per 100 horse years at risk. You can see that's down a quarter of what it should have been on these premises. Maybe that's a good thing. But what was interesting is that on those same premises during that period, there were 16 additional grass sickness cases amongst horses that had not been enrolled onto those premises. So grass sickness was still occurring, and it was still there. Those animals we know were not vaccinated, but we can't include them in the data analysis. If we could, you could argue that the answer is there. But these animals were not eligible because they were not part of the trial. So we had a unique randomized clinical trial in equine medicine. It was large scale, it was multi-center, it was consortium funded. This is my last slide that I'll go to, and you'll see the number of groups and individuals that contributed to this trial. It was a large trial, and all credit goes to Joe for sorting that out. There was a very long recruitment phase. We now know from human studies that organisers of these often overestimate the time or underestimate the time that it will take to complete them. And indeed, we probably fell into that trap. There was a greater loss to follow-up of these animals, more movement of animals than we expected. That did lead to a longer trial period, and it did mean that more animals moved around. So it became a bit of a self-fulfilling thing. I think the trial methods were actually successful. We'd taken them from human methods, and we managed to identify high-risk premises. We had absolutely excellent compliance from owners and their vets, so no complaints there whatsoever. We did conduct some interim analyses looking at whether or not our randomization was working okay, and that certainly was true. We've done that twice during this. And it also confirmed that we were getting very good vaccine safety as well, so nothing to be concerned about there. But the low incidence, we had low numbers of cases being reported throughout this trial. And that did mean we had to extend this trial by 18 months longer than we had intended. We had to reapply to extend our animal test certificate with the Veterinary Medicines Directorate in order to complete this. As you've seen, not all horses and ponies on each trial were enrolled, and maybe that's something we need to learn from. And we did end up, I think, with a proportion of these animals in that sort of older age group on these premises than we perhaps would have ideally liked. And it is known that contacts of cases do tend to be better protected as well. So perhaps that was all contributing to why we had low disease incidence. So the work is going on. We have got all the data in, scanned, verified, collated. The serological assays, testing those blood samples for antibodies, is now completed, but only fairly recently. We are conducting these, what we call multivariable statistical analyses, taking into account other factors that might contribute to this. And once we've done that, then we will be lifting the actual blinding. But I just wanted to end by, I think, giving you some hopefully encouraging evidence from this trial, and it's really just confirming what we saw in both the safety and the pilot. And as I've said, we consider antibody responses expressed as the ratio of the post and pre-vaccination lives. So essentially just dividing one figure by another and looking at that ratio. Where we have a ratio of 1, that's no effect. Well, these are the data now from this trial, and you can see that these are discriminating quite nicely. This is where the ratio of 1 is, and we see a cluster here. The rest of them are shifted over here, so positive ratios. We can express that in a slightly different way by looking at the distribution here of the frequency, and you can see that this red group here, they center on 1. The blue group obviously displaced down here, so they've got, on average, higher ratios. But that, to me, says something is going on with this product when we put it into these horses. But there is a but. And that relates to the slightly darker shading here. And this is where these two distributions overlap. And this is the problem. We would have animals with antibody here amongst the non-vaccinates, and equally we would have some vaccinates that sit nicely with no evidence of antibody, or at least no evidence of response. And that presents us with a bit of an issue. And so really the last slide that I really wanted to just say was there's always a but, and I think we do need to be cautious even when we're assessing non-significant results. You remember that I mentioned we had a pilot study, a limited number of animals on a limited number of premises. That study was not set out to test this vaccine, and we had one case of grass sickness while that study was being conducted. When we broke blinding on that study, we found that that was actually in a vaccinate. It was an animal that had received the toxoid. And what we found was that actually that was what we believed to be a poor responder. That animal only had a 1.2-fold change from its vaccination after receiving 3 doses. And if we look at its position on this distribution, this is the placebo, that sharp group there, this is the vaccinates. This animal sat there right at the interchange of where placebo and vaccinates couldn't be discriminated. So we think that animal was a poor responder, and we may well have those in our set of data. So analysis of the vaccine effectiveness must control for how these animals have responded to this vaccination. If we don't do that, we may be coming up with the wrong interpretation of our results. So finally, I just wanted to really acknowledge all what I've called the facilitators, the people that have been responsible for conducting this, especially Dr. Joe Ireland, the principal investigator, but many other people have been involved in facilitating this. And then lastly, that funding slide, all of these funders contributed, some of them quite considerable amounts of funding to make this happen. And without all of their contributions, this trial would never have got off the ground. We've got a little bit of a way to go just to finalize the analysis, and then once we've done that, we'll be able to release the full results. So thank you very much, James.